Alright, welcome back everybody. This is the Principles of the Innate and Adaptive Immunity. This is chapter 1.6 on Lymph Node Anatomy. So make sure to hit the subscribe button and follow along because this will be the first lecture of 3 different organs that we'll be talking about in the lymphatic system. Next one will be the spleen, and then following that will be the Peyer's patches. So this is epitope immunobiology. Let's begin. Okay, so lymph nodes, okay, they are oval-shaped organs that are the major site of B and T cell maturation. Okay, they exist primarily around armpits and the stomach lining. And what we want to talk about here is that there's a diagram here of what a lymph node is created. So let's follow along. The lymphatic system is what connects all these lymph nodes. Okay, and lymph nodes is really the major portion of the lymphatic system. So it's an extensive system that collects extracellular fluids from the tissues and then returns it back to the blood. Okay, this extra fluid is called lymph. Okay, that would be very, very important. It's most basically everyone calls these lymph, and it's produced continuously by filtration from the blood. And we're taking essentially lymph or these extracellular fluids from the blood that's collected from the tissues of the body. Okay, so lymphatic vessels carry lymph away from the peripheral tissues. So it's very much like a blood vessel where you got an organ, and then connected to the organ are blood vessels that basically provide oxygen and nutrients to this tissue. But in this case, they're supplying everything that's regarding the immune system. So lymphatic fluids. They're very much like blood vessels as well because they're unidirectional due to valves. So like the blood system where you got a valve and then you just got a blood vessel and you got a valve so that the blood can only go one way. Okay, it cannot go the other way. Okay, that's not allowed because these valves actually protect it from prevent it from flowing backwards. And afferent lymphatic vessels are those that carry lymph away. Okay, remember this is away from infected tissues to lymph nodes. Okay, so this is from the tissues to the lymph nodes. Okay, that's what the afferent is suggesting, afferent lymphatic vessels. The other type of lymphatic vessels is the efferent. Okay, so the efferent lymphatic vessels, as you can guess, carry lymph away from the lymph nodes then to the tissues. So this time it's lymph nodes to the tissues. All right. So essentially, if this is the lymph node, this would be the afferent. Okay, so it's carrying from the tissues, and it's got a T here, into the lymph nodes, and then this would be the efferent vessel, carrying it out, okay, and back to the tissues. All right, so what we have here is the lymph node. I'm going to go through exactly how this works, starting from the afferent vessels, lymphatic vessels. So you see here you got the tissues. Okay, I'm just going to put tissues here. It drains the lymph into the afferent lymphatic vessels, and they bring them to the lymph nodes. Okay, and then from there they go on into these what we call cortical sinuses. Okay, so you can go here, and of course the cortical sinus are labeled here. It would be in this way as well. Okay, and it goes into the cortical sinuses, and they can actually go 2 different ways. Okay, the first when it gets here, it can go into this blue area. Okay, called the paracortical area. Okay, paracortical area is to consist mostly of naive T cells and also activated T cells. Or it can go into these medullary cords, okay, where there's macrophages and plasma cells. So these areas are especially important because they express chemokines that essentially are chemoattractants that then bring antigen-presenting cells from the surrounding tissues, okay, to the paracortical area and the medullary cords. And so essentially, if there is naive T cells here, okay, in this area, and they're expressing these chemokines, and they basically tell these antigen-presenting cells, for example, the dendritic cells or macrophages, to go into this area, they can then bind to all the T cell receptors, cause them to activate and then become effector T cells, which would be, again, helper T cells, cytotoxic T cells, or regulatory T cells. Okay, so this is where a lot of the T cell maturation will occur. So that's what mostly T cells, and of course, maturation occurs here. Maturation for T cells, of course. All right, and then when they're in this area, they also are actually going to bind and activate in this kind of lining region right here that's touching the, okay, we'll call them primary lymphoid follicles, or this area is called the cortical secondary lymphoid follicles. Okay, this is the germinal centers. This is primary because there's no yellow dot here yet. But as this germinal center then begins to grow and grow and grow, so you see you got a lot of dots here. Then they make like say a germinal center, and it gets bigger and bigger and bigger as they continue to proliferate. But what happens is that these antigen presenting cells then are binding with B cells in this region. Okay, oops, my apologies. So they're binding with these B cells in this region here, and they actually, as I mentioned earlier, I mentioned that the B cells don't just activate due to binding with antigen presenting cells, but they also require help from helper T cells. Okay, so the helper T cells that were activated in this region, okay, this paracortical area, can of course also participate in the binding of B cells, naive B cells. And then once they of course get all the significant signals to then survive, proliferate, and differentiate into effector B cells, which we would call plasma cells, then this is where all the proliferation and essentially differentiation begins. And it gets bigger and bigger and bigger and bigger to make one full germinal center. Okay, and then once they're completely grown into effector T cells, and there's effector T cells, effector B cells, they essentially are ready to leave, because this is essentially, it was a naive army of T cells and B cells that are now armed to target the enemy, which would be an infected tissue that came from the outside. So once they are differentiated, they actually downregulate certain cellular receptors, so they're no longer attracted to this area, and then they can then leave. So once they're growing, say this is completely grown area, and you got a lot of plasma cells, okay, plasma, my apologies for the very, very ugly writing, or the effector T cells, let's put T cells here, then they can then leave, okay, they can go through this area because they're no longer homing in this area, and then actually exit through the efferent lymphatic vessels. Okay, so they go this way, and then this will go back to the tissues. Okay, and remember that there's a lot of cells that are expressing cytokines and chemokines in this area where there's inflammation, and so therefore these cells are now going back to the area of infection. Okay, so I'm going to put infection here, and this infection then is now can now be targeted by B cells and T cells that are specific for the antigen that was brought from that area earlier. Okay, so if there is an antigen that was brought back on MHC from these antigen-presenting cells earlier, so dendritic cells, for example, they go to this area, then they tell, so you got this triangle, they just tell these T cells and B cells to target this triangle, okay, because this triangle is a signal for a foreign infection from that area. Now they know all of them, once they've essentially gone into effector T cells, they know to target this triangle, and so they go back out, home back to the area of infection. Now they know to kill that triangle with an adaptive immune response. Okay, and that whole process takes about 6 to 9 days. Okay, 6 to 9 days. Okay, and that's why often when you get infected and you get sick from 6 to 9 days, because your innate immune response couldn't sufficiently fight off the infection. But then the innate immune response with the dendritic cells and macrophages, so on, bring it to the lymph nodes, activate the adaptive immune system, and then this process takes 6 to 9 days. Then mounts a very, very immense and strong adaptive immune response of T cells and B cells to then go target that infection. So then you can feel better after about a week and a half. All right, and one other thing to mention here that you see I've labeled here, arteries and veins. These also carry B cells and T cells that are naive, that are circulating in the system to the lymph nodes. And so what happens, like, there's actually connected through a very, very complex network of capillaries, but actually they are connected to something called high endothelial venules. I'll show you that in the next slide. But then these areas where there's the macrophages are expressing a lot of chemokines and cytokines. Essentially, the chemokines attract these naive T cells and B cells from the artery to the medullary cords, and of course to the paracortical areas, because now they know that there's an area of infection, or not infection, but rather there's an area with an antigen presenting cell. so that we can now become effector T cells. So they must actually be drawn through the arteries and veins through high endothelial venules that enter into the medullary cords. All right, so a quick summary of what that was. So number one, the afferent lymphatic vessels drain fluid from the tissue and carry pathogens and antigen-presenting cells bearing antigens to the lymph nodes. All right, so I mentioned earlier that goes to the afferent lymphatic vessels. Number two, the dendritic cells are secreting chemokines simultaneously and attract the naive lymphocytes from the blood and enter blood vessels through specialized blood vessels called, I just mentioned, high endothelial venules. Okay, HEV, that's a very, very common acronym for high endothelial venules. And in the lymph nodes, B cells make up the outer cortex. So this is where the germinal centers were, while the T cells are more diffusely distributed in the surrounding paracortical areas. Okay, so that's the blue area that I mentioned earlier. They also call this deep cortex. If you see this anywhere else, that's the same name, deep cortex and paracortical areas, or T cell zones, because they're primarily composed of T cells. The free antigens / pathogens also are able to migrate to lymph nodes where they are caught in the paracortical area. So I didn't mention this earlier, but essentially, if there happens to be a free pathogen or just a little piece of a pathogen, for example, an antigen that just happens to float through the blood system, they can also end up getting caught into these lymph nodes. And so they get caught in these paracortical areas. And then, of course, the dendritic cells can process them and so on and expose them to for adaptive immune responses. So we got naive T cells and B cells that successfully interact with antigen-presenting cells. We'll go through clonal expansion in the germinal centers, and B cells, again, will require assistance further from helper T cells. I mentioned earlier, not just an antigen or pathogen will be enough to activate a B cell. You also need the helper T cells. All right, that's very, very important. And lastly, effector lymphocytes may then exit the lymph node and migrate back to the area of infections through efferent lymphatic vesicles, and this is directed by cytokines. Actually, chemokines, okay? Cytokines and chemokines. That's really important because they're the chemokines, because they're the chemoattractants. All right, great. So that's a quick summary of how lymph nodes work. Next, make sure to follow next time on chapter 1.7. We'll be talking about the spleen anatomy, and it's very similar in how the spleen works, but essentially they have a slightly different function. But we'll essentially talk about the principles and fundamentals of how a spleen anatomy works and how this contributes to the adaptive immunity. So make sure to click on this link right here, and of course follow along by subscribing and hitting that like button. If there's anything you would like me to clarify or even make a lecture on, please comment below. So this is Epitope Immunobiology, and we'll see you next time.